thank you for the organizers for setting up this conversation. And it's nice to be back in the Newton Institute, and thanks to the institute itself. The professionalism of the organization is always impressive here. So there are a few things I'd like to share with you. So this might be kind of a slightly a fast talk, because there are too many things I'd like to share with you. I'll plant the ideas, and perhaps we'll have the time to talk about them later. Of course, we can't be in the Newton Institute and not use the chalkboards. This is just would be a crime. So, the to-do list or the open questions, we'll just have to put them there, just to use the facility. Yeah. So as I go along, we'll add to this list. Right, I'm talking on behalf of a number of people. The clever questions should go to my colleague Alejandro Einstein, sat there. So he will give the clever answers. So we're kind of, would like to kind of present the group just to give you a context of where we're coming from. So the Brain Dynamics Lab is actually a virtual lab. We're distributed around about 4 or 5 countries. And we're split between people who work on kind of ideas from physics, maths, engineering on one side, and the experimentalists on another side, using kind of EEG mainly, but we also use fMRI. And the idea is really grounded in the maths, the engineering. So we take a dynamical systems approach to brain function, and we use that to try to make interventions. So the question we're trying to plant in here is, how do we use that conceptualization of brain dynamics to inform neurorehabilitation? So the question there is, so now we have multiple of meta-analyses that show that electric stimulation has a role in improving cognition. But people say the results are very variable. Why are they variable? And why is there no protocol to inform how we actually implement neurostimulation? There is no established protocol. And maybe the fact that there are no established protocols explain that the variability that we observe in the data. So can we actually use some of the ideas that we have here from maths, physics, maths, physics, and engineering to design better protocols for neurostimulation? The work that we'll be presenting here is thanks to a large group of people, and I will be mentioning them as we go along. So this is kind of the basic summary of what I'd like to talk about, kind of the idea of a dynamical system approach to brain function, the emergent properties that come from there, and then this idea that neurostimulation is a control over the dynamics. How do we use that for personalized neurorehabilitation, and think about stimulation as a network intervention? So a lot of our colleagues would use neurostimulation, electric stimulation, to say we stimulate the prefrontal cortex. Well, you're not. You're stimulating a network of which the prefrontal cortex is a node. And that conceptual change needs to be stressed. Right. This is kind of our mantra slide. So, and I'll try to kind of support this as we go along. So we say a behavior, a task, a cognitive task, behavior requires resources. Brain resources are brain networks. Those networks will have a fingerprint given by its oscillation spectrum. In such a way that if I can intervene on the oscillatory properties, I can favor the network, and as such, favor the underlying behavior. And I'll try to support this as I go along. But this is really my base camp slide. And I will repeat presenting this to cover some aspects of what we will be showing in this aspect. So the first start, I'll show the relationship between the network and the spectrum. And in that, I'd like to plant the idea of network resonance. In the same way we relate it to physical objects. Oops. How do I go back? Yeah. So we have no problem kind of relating the concept of resonance frequencies for different size instruments like this. I don't think anybody here would dispute that. But it seems that the idea of saying networks would have a similar property such that a small network will have a higher resonance frequency than a large network. That seems to kind of, with physicists, seems to kind of jar a bit. So we'll try to support this. So the work really started from the PhD work of Caroline Leah Carnell in Manchester, who had beautiful demonstration on using experimental, very elegant experimental. I have to say that Nikos Ligoussis planted it for us, kind of modified it for us when we started, and a model that shows that there is an inverse relationship between the size of the network and its favorable frequency. And this came from an observation that people, somebody mentioned the steady state response yesterday. We observed this experimentally, that if you stimulate sensory systems, auditory, visual system, with a rhythmic stimulator, flash, or a series of clicks, or a tactile, repetitive tactile stimulus, you'll get the same frequency. This is the entrainment effect. You'll get the frequency of the stimulus in the brain. And we use this to track attention and so on. But there is a favorable frequency for each of those sensory systems. So we know that the favorable response for a flash repetitive stimulus in the steady state is around the 10 hertz frequency. Whereas the auditory response is favorable, the maximum response occurs around 40 hertz clicks. And the question that Caroline planted is why? Why? And the hypothesis was that the response is inversely related to the size or the volume of the sensory cortex that's being stimulated. And she shows that very elegantly. However, our colleagues in physics, and based on that, she shows some work and did some work. I'm using this as a tangent to highlight her work related to frequency-dependent plasticity. So this is a point here for discussion. Frequency-dependent. And the plasticity aspect I'll come back to later. So she does a very elegant model, and she does show that experimentally that there is a relationship between the resonance frequency and changes in GABA concentration. She measures GABA and verifies that with a model. Our colleagues in physics say, well, no, this is not true. It can't be true. We have at least 3 papers that show this effect. They say no, because Steve Strogatz didn't show it. In Steve Strogatz's book, he doesn't relate anything to network size. So we went around and did the analysis. And this paper is now under review so we sent it to physical review letters, and they said, it's too short for a letter. Send us the full paper. So it's now under review in physical review E. And the analysis was basically done by Felipe Torres. And we used the formulation with the Koromoto models, a model of loosely coupled oscillators, to allow us to do this analysis. I'll come back to the Koromoto as a useful tool to analyze whole brain networks in a bit. So in Koromoto, we have a dynamical system, an evolution of phase over time, that is a function of local properties, intrinsic properties, which is an intrinsic frequency of a node, and what it receives from where it's connected. So it has a connectivity weight matrix to its neighbors where it's connected, and it has a delay. So the length of the fiber will change the time it takes to transmit the signal. The delay is important here. And then we can control it overall with a kind of a global connectivity parameter that scales the whole thing up and down. And we'll see that this is useful later for establishing its metastability.
It's a fully connected network?
In this case, it's fully connected network because it facilitates the analysis.
But if you just not fully just cluster. It's maybe not the size of the network.
Yeah, but this is fully connected. So when I use this tool now to later to model whole brain networks, it will not be fully connected because it will be informed by the connectivity of the brain. Yeah? So for the purpose of the analysis, this is fully connected to also make the analytical solution tractable. And then we say that near synchronization, we can make a few approximations. When it's nearly synchronized, I can drop the sign. I can also approximate that the phase is a linear function of time, another simplification. And then I can then reorganize this and write it in a matrix form where I can then solve for the emergent collective frequency, and it turns out to be a function of where the number of nodes become part of the equation. And if you put it into the limit, it converges to Steve Strogat's equation. So Steve Strogat says that he doesn't actually have delays in his model. It converges to the intrinsic frequency. So wherever you start, random phases, the system will converge to the intrinsic frequency. Ernest Neuber showed later that it will converge to a frequency scaled by the delay, so by this tau. Yeah? So we can show that we converge to Neuber equation with homogeneous delays. So there are three cases where we can highlight how does the network scale as we grow it. So we can think about it from the point of view of physics. Think about physics. If we have a volume and we add more particles while fixing the volume, it increases the pressure, right? Basic physics. If you pack more particles in the same volume, it increases pressure. So that's like increasing the connectivity weights.
If you don't change temperature.
If you don't change temperature. If you don't change temperature, thank you. Yeah, so I'll fix the temperature. I'll fix the volume, so the pressure increases. That means it's like the connection increases. Likewise, if we inflate the volume, so add the particles by inflating the volume, it's like fixing the connection, the pressure between them will remain the same, the volume increases, so delays are increased. Yeah? So there are two ways of seeing this. Yeah? So these are the two ways. And you can see that the collective frequency that emerges from the interaction goes down, scales down, as the network increases. So the resonance of the system, or the favorable frequency of the system, or the collective frequency that emerges from the interaction of the oscillators, goes down as the network increases. And this example is kind of like playing with both, and this was added at a request of our colleague Peter Robinson in Sydney. Right, so we have a proof, a kind of analytical proof. I hope this will satisfy our colleagues in physics. Well, what does that got to do with the brain? So if we can see now that in the brain we have a number of interacting networks. Networks will have their fingerprint, their oscillatory fingerprint, and if I can intervene with this oscillatory fingerprint, I might be able to favor the network. It's just like kind of now tuning a guitar. So it doesn't, however, explain, this proof does not explain why, in the case of the EEG, we get this discrete bands of frequency modes. Because I have resonances, I have highly connected system in the brain. Why don't I get a kind of a continuous frequency of oscillation? Why does it come in bands? So we go back to the Koromoto and try to kind of inform it a little better. So this is the work of Filippi and Monica Otero. So we go back to the Koromoto and we say, okay, we want to explain the EEG. So my level of analysis is the EEG and the spectrum of the EEG. So I'm interested in oscillations. I'm interested in oscillatory modes that are generated by some underlying interaction in the brain. So starting from the level of representation of oscillation is not a bad idea. Where do they come from? We can have that as a different discussion. That we can model. Janssen and Ritz is a good model. Wilson-Cohen is another model that we can generate from that the oscillations. But it's a reasonable assumption that we can start modeling the EEG as a series of oscillators. And the oscillators here come from a parcellation of the brain, in this case a very rough parcellation. There are 90 nodes here, the AL90. I'll insert an oscillator in the centroid of each of those nodes, and then I will connect them based on the structural connectivity information that come from the connectome, for example, from the Human Connectome Project. And it comes as two forms: the connectivity matrix, the adjacency matrix and the weights, and the delay matrix, the length of the fiber. So I have the intrinsic local information, intrinsic frequency, which I can play with, and the global interaction across the system. The matrices come from our colleague Pamela Guevara in Concepcion, and they do beautiful work in actually calculating deterministic and probabilistic connectivity matrices, different techniques, and they develop these techniques for us. So what happens if we then run the system, and with one set of parameters, so the three parameters I can play with are this K, the global coupling that I scale up the system up and down, and some scaling of this delay. These are the only two parameters I can have. So with only one set of parameters, we observe the emergence of metastable properties, like what we observe in the EEG. So all of my nodes start with intrinsic frequency at 40 Hertz. All of them start at 40 Hertz. And I get a full spectrum. Now, the model does not generate the 1 over F. So I get a full spectrum. In this case, it's showing 5 oscillatory modes. And it shows that we color them here, and we color their time domain. So you see, these are nodes, brain nodes. And you can see that the nodes switch between the oscillatory modes. So they occupy a certain duration, one mode, they switch to another, and then they switch to another. So the durations are of interest here. Which frequencies they are is of interest. And if I analyze their functional connectivity, we can see that these are the dominant networks that emerge. And again, notice that the lower frequencies produce larger networks, coming back to the resonance property. Yeah? Whereas higher frequencies produce more smaller local networks. I mean, we know that from the EEG literature. 40 Hertz, the gamma band, is about small local networks, whereas the lower frequencies, alpha, theta, are about global connectivity in the brain. Right.
Sorry, this is data or model? Because
this is the model. All of this is modeling.
Why are you labeling it on the left?
Because I'm using parcellation. I'm using a real brain parcellation. So these are brain areas based on an anatomical parcellation that I'm connecting them according to the real structural connectivity. So the real data is the atlas and the structural connectivity. Yeah?
Like a synaptic matrix, yeah? Like a synaptic matrix.
See, the white fiber tracts. So, I mean, at this level, I can't use the word synaptic. I'm not allowed to use the word synaptic. I'd like to use the word synaptic. I'm not allowed to use the word synaptic. These are white matter tracts.
Okay. So why not being allowed? I mean, there obviously are white matter tracts which synaptically connect different areas of the brain, right?
Yes, but that jump apparently is I cannot make explicit. Apparently.
But why? That's why I'm not understanding.
Yes, I mean, it's important when I come to talk about plasticity, because one key element here is going to be this is a dynamical system that you'll intervene with, and it's an adaptive system, so it will change. What changes? Now, white matter tracts, we observe changes on the stimulation within half an hour. White matter tracts do not change in half an hour. This is why.
Sorry to interrupt as well.
Yeah, please.
So in reality, these dynamics are generated from functional connectivity or effective connectivity. So why do you use structural, and how would it change if you use information?
You don't get the spectrum. This is a very, very important question, because this spectrum is functional structure. I mean, notice that it's coming in banded. This is why we use the word oscillatory modes. They are resonances of favorable or dominant functional networks, but the functional networks emerge from the structure. If I change the structure, so your structure connectivity matrix will be different from mine, we'll get a slightly different profile. The dynamics is the same, the system is exactly the same. By small variations in connectivity between us, we'll get a different dynamic. There first.
So this is quite interesting. Now I am referring specifically to 40 Hertz auditory steady-state evoked potentials or 40 Hertz steady-state visual evoked potentials that have been obviously described in the literature extensively. And it is pretty clear that somehow there has to be a resonance in the system when you stimulate it. What I personally can't wrap my head around is that in contrast to lower frequency endogenous oscillations like alpha or something that we've talked about, they have a certain frequency that's different in different people. And you can sort of see how this is some thalamus-cortical interaction or something. The higher the frequency, obviously, the smaller as you, I think, said, the smaller the structures have to be. How is it possible? So I guess there's two questions. Like the lower frequency oscillations, they differ in people. They differ from structure to structure. Like visual alpha is different in frequency from the sensory motor rhythm, for example, in frequency, and they are different across people. However, like a 40 Hertz steady-state visual evoked response will be exactly 40 Hertz, not 41. It will be 40, and it will be 40 all the time in different people. So I can't wrap my head around how that is really possible. I mean, one could maybe say that the 40 Hertz comes from some interaction between excitatory pyramidal cells and inhibitory interneurons, maybe because they're pretty close to each other, and so maybe a 25 millisecond, or something latency would make sense. But it just that the fact that we get a very, very coherent and strong response to a stimulus, and that is the same across people, and is really exactly at 40 Hertz, is something that I don't understand.
Maybe a few more slides on it answers that question, because I mean, once I haven't stimulated the system yet. So once I show you some,
it's just resonance frequency.
Yeah, this is just the intrinsic, the metastable intrinsic dynamics with one set of parameters. I'm insisting that I'm not changing anything here. The system is generating this by itself, yeah, and it oscillates, it moves, it switches, it transitions between the different modes automatically. When we stimulate, we'll intervene with this, yeah. Any other burning kind of points, Francesco?
Yeah, sorry, just to go back 1 second to the fact that white matter connectivity doesn't change within 30 minutes. I mean, that is true structurally, but functionally speaking, you do have, at the point of connection, wherever it is between the two areas, the presynaptic and the postsynaptic points of exchange of information, they can indeed change the efficacy in transmission of information within 30 minutes. So you don't see structural changes in the white matter.
So what do I need to measure to show that? What do I need to measure to show that?
Well, I mean, in brain slices, it's synaptic plasticity, right?
Yeah,
but here it could be very well this change in oscillation.
We see the change in oscillations. We see the change in the functional networks. but we don't, if we make the mapping, the direct mapping between white matter tracts and synaptic connections, it's not that straightforward because of this timing issue. I mean, can I carry on and then maybe we can come back to it? But it is really an open question, because not only that this plasticity, we show this plasticity depends on the frequency of the stimulation.
Yeah.
Right, so what we can do here, we can do interesting things here with this now. So instead of one set of parameters, I can then sweep across my 2 degrees of freedom, the global coupling and the mean delay. I scale that and I can look at what kind of spectra are emerging from this. And I can measure some sort of metastability. Here we measure it by spectral entropy. This is a point of maximal spectral entropy that gives me the maximum width of these oscillatory modes. But I can do other things now. I can then project the real EEG data onto this space by looking at similarity between the EEG spectrum and the spectra that come at each point in this map, in this heat map. So the color only reflects the metastability, but I can relate real data. So, for example, here is a square where it is a young, healthy person's EEG, quite close to maximum metastability. Here is the point of data of a healthy elderly person. Here is a person at sleep. Here is a person in deep anesthesia. So I can relate real data to the parameters of the system. So I can now, although I'm using the connectivity matrix of one person, or the median of a population, I don't have the structural connectivity matrix of every person. But I can make a similarity or a dissimilarity of the data from real people onto the parameter space within the model. We'll see that this might become of interest later, because then the task might be that I want to move somebody's spectral properties to a different place. So somebody who has, in this case, an elderly person, I want to improve their spectral properties to bring it closer to maximum or better metastability, because we suppose that this is better for cognitive flexibility, for example. Yeah? So the task would be, how do I make, now it becomes a question of control. Here's a kind of a very simple example to highlight this point. I can now also use the system to simulate disorders. Here is a simulation of a stroke. So this is the coordinates of a real ischemic lesion that came from our colleagues in Bilbao, and we can then damage the structural connectivity matrix with this information and see what the spectrum would look like. I mean, patients with stroke, the clinicians wouldn't allow you to touch them. So I can't actually get EEG from patients in an acute case, because I mean they're trying to save their lives, so they wouldn't let me. So one way is actually be able to simulate their data, and this is interesting because we know that in many stroke patients we see this shift, a kind of increase in the higher frequencies and a shift in the lower frequencies. So it's kind of consistent with what we observe, right? Yeah. So one point that relates to somebody raised the point yesterday, and I think it will come up again. Okay, so we have this the non-stationarity. I think you raised it yesterday. So we have this point of, we have a mode, a frequency mode that then stops, switches to another mode. So there's clear example evidence of the non-stationarity of the brain dynamics, or at least in the model. So how do we estimate this dynamic? Again, I'll make a tangent here to say something about our work developing models to estimate brain states. So the idea that here we can see that these modes are discrete, emergently kind of discrete. I didn't specify them. So we can think of a model where I have discrete states emitting continuous data. This kind of calls for something like hidden Markov models or hidden semi-Markov models. So here we kind of use the idea to think about the networks, because the oscillatory mode reflects the network. That's according to the mantra, network, oscillation, behavior. So I have a series of networks that switch over time and they emit the observable. And here is a kind of series of papers where we develop methods at the level of the time domain at the level of the spectrum, at the level of the network itself, to estimate those states and their properties, like transition probabilities, durations, etc. And a couple of papers to use these techniques to say something about EEG. Like, for example, what does the resting state alpha tell us about the propensity of some people to hear voices? So there is no task here, and there is no diagnostic, because there are no patients, but is there something about the dynamics of the alpha, the trajectory that the alpha takes, to say something about those people's propensity to hear voices? Right. So I move on and say, right, so I have those modes, and the question becomes naturally, like, what happens if I now drive the system at one of those modes? So I externally drive the system with something like an oscillation at one of those modes. Will I then favor the underlying network? Will I then generate more activity in that underlying network? So this is the work led by Monica Otero. And we have a number of examples. So the paper is now, we have the paper on bioarchives, and it's now under review in PLOS Computation Biology, I think. So she shows kind of highlight examples. So she stimulates the right precuneus at 3 of the dominant modes that we have, kind of low, medium, and high frequency. She uses here the precuneus because it is the node with the highest node degree. It's the most connected node. Fine. And you can see that stimulating the same node at different frequencies activates different networks. So there is a mapping between the frequency of stimulation and the network we activate, coming back to the mantra, network oscillation. I drive the network at different frequencies, I favor different networks. Again, lower frequency, larger network than higher frequency. She changes to a mediumly connected node, so I change the position, so same frequency of stimulation, different node, different network. Again, this is kind of goes contrary to this idea that I stimulate the prefrontal cortex at theta to improve blah. Yeah? Stimulating the prefrontal cortex is stimulating a node in a network. And depending on the frequency, you will be selective of that network. So, and then the least connected node, the paracentral lobe, it's not globally connected. So there is no transmission for lower frequencies. And only when you stimulate at a higher frequency, you generate a local network, as we'd expect, a dominated by 40 hertz local network, consistent with what we observe in the data. Coming back to your question. So I'll now show a result with it directly. So before I show that, very quickly, because I'm running out of time. So the interesting point here, this is now some work from Felipe, where he stimulates across the board. So in here, we stimulate the modes. Here, we stimulate all the frequencies. And it shows that...
Sorry, is this all in modeling?
All of this is modeling. All of this is modeling. When it's real data, I'll tell you. I will show real data. It's simulation, but what's real, I insist, the connectivity matrix is real, is a real person. Yeah, so if we see here, on the x-axis, there are brain areas, on the y-axis are stimulation frequencies. And you see that the red dots is activation of the corresponding nodes or networks. And you see that if we vary the stimulation frequency, we only get responses, or we get strong responses, if we're stimulating close to one of the modes of the system. If we stimulate far from it, like I'm stimulate around here, we really don't get much. So stimulation needs to be close to an already existing one of the favorable modes of the system. And the next slide shows that mostly you get an increase in the network activation. You get some decreases. But so stimulation at the mode frequencies favor the corresponding or the underlying network, which is the point I was trying to make. And finally, kind of simulating the steady state effect. So here we stimulate 2 nodes that correspond to visual cortex and auditory cortex. And we do it in 2 runs with parameters as if we're simulating 2 different people. So those are practically two, by using the projections of the EEG onto the parameter space, I can use metaphor of simulating people. So those are 2 different individuals. And if I stimulate the visual cortex at the 3 different frequencies, you notice that we get slightly different networks, but you get maximum response in the visual cortex at the low frequency. So while you will get a visual evoked response at 40 Hertz in everybody, because whatever, you will always get the 40 Hertz, which is you will always, if you have enough energy, you will get whatever stimulating frequency. If you stimulate at 41.1, you will get 41.1 in everybody, if you drive it sufficiently. So we drive it here at 43, you still get a network. Yeah? But the maximum response in the visual cortex is in the alpha range. Whereas in the auditory cortex, the maximum response in the higher frequency, which is what we see in the auditory steady state. We get a favorable response between 38 and 42 Hertz. But if you drive it at 50 Hertz, you'll still get a 50 Hertz if you have enough energy. So we showed this in the work of the PhD of Monica Otero. David Ollier with Carl Friston showed something similar with this when they used an extension of the Janssen and Ritz model. Right. I was going to leave this and the few next slides now when I have a 5-minute talk when I talk about the poster, so perhaps I'll leave it there. But this would be the answer to your question. Because if you have sufficient energy, you can basically get whichever frequency of stimulation you like. I'll skip the next slides just to get us to real data and leave this to my 5-minute presentation of the poster because some of this is already in the poster. So in summary, stimulation frequency activates a network. So when you talk to your colleagues who use TACS or TDCS or sensory stimulation, please remind them of this fact, that we need to think about stimulation of networks, not nodes. You can favor a network by driving it close to its oscillatory mode, and you will favor a network by driving it at its mode. Let's look at some data. So based on this concept, Alejandro had a very nice project working with a mining company where people work at high altitude, and there's all the problems with high altitude. So he showed here kind of a device that did the tactile stimulation. So they didn't want anything auditory or anything visual because that distracts them. So Alejandro invented this bracelet that drives tactile stimulation at the resonance frequency of the somatosensory cortex, around 23 Hertz, and shows that when people come from sea level to high altitude, 4,000 meters, and they do this in 2 hours, you get this disruption in cognitive function, which continues, whereas when you use this device, it stops the deterioration. It kind of maintains the performance for longer. And I think it's gone to a company now. There is a patent that came out of this, and there is a product. So you can actually use these concepts to actually produce something impactful, useful. Going back to the mantra, I just want to show you some evidence from data that has just come out. So this is the work of Andre Gomes. So he showed something, started with kind of a group of younger and older adults, healthy. They can do the task, they do the task well, but he relates their task performance. This is a very simple kind of solving a conflict. So you press the button with the right hand, but you're given the instruction in the left ear. So that kind of gives you a time to resolve the conflict. And he shows a relationship between the task performance and the frequency of the intrinsic frequency of the individual theta frequency of the participants. So we usually, in analyzing EEG, we use the power. And if you analyze the power, you'll get differences there. But the elegant thing about Andrew's work is relating it to the frequency. So he shows here that people who have higher theta frequency have better performance. The lower here is better in the sense that they were faster in resolving the conflict. So, and the fact that younger people have higher theta frequency, we know that as well. So we know that with aging, the EEG spectrum shifts to the left. There is a slowing down of the frequencies. So that's typical. So this is dominant here. Older people are heavier on this side and younger people in here. And then he relates that to connectivity. I'm not going to show this result, but the question arises, given the information I showed you now, it would make sense to say, right, if I then try to shift the theta frequency of those people slightly higher, I should get better performance. Is that right? If higher theta oscillations correlate with better performance, if I now shift the people with lower frequency, I should improve their performance. Yes? No? Maybe. But at least given this narrative, that would be kind of a logical thing to do. So that's what Andre did. And what he did is with auditory stimulation. So he delivered auditory clicks at a low frequency common to everybody because they're older people, they all have low theta frequency, at the individual theta frequency, at a slightly higher than the theta frequency, and at something completely random, arrhythmic, but counting for the number of stimulations, to balance for the number of stimulations. And then he shows that there is entrainment, that when you deliver fixed frequency, you get the steady state effect, and that people who have poor performance to start with have a higher increase in their performance if you stimulate them at the individual or slightly higher than the individual frequency. So this is about personalization. So in TAX, the standard protocol is to give the same frequency to everybody. We're saying you need to adjust it to everybody because the frequency relates to a network. And what you want to activate is the network of this person. So you need to stimulate the personalized network of this person. So you need to find the corresponding frequency of this person. Daniel?
We have troubles with Spanish, but that means the
Sorry, it's in Spanglish.
That is the percentage change in performance.
Yes.
It's not relative. It's not absolute. It's relative.
No, it's the percentage change compared to the baseline.
To their own baseline, yeah?
Yes.
That's very sizable, in fact.
Yes.
Very sizable.
Yes. So I have 40 seconds.
Sorry, go on.
So and the boss is there. So the point is, there is personalization matters because I wanted to show very quickly this as well. This is related to electric stimulation, personalized electric stimulation in people with MCI. And here the task, it's a memory task, and here it favors lower frequencies. So theta frequency, but lower frequencies. The details I can show separately. So here we gave them either 5.5 hertz, it seems to be common in many of the memory stimulation literature, higher frequency, 8 hertz, or we gave them slightly below their own frequency. And when we give them slightly below their own frequency, we get a better performance. That's the summary, and it's run out of time. So thanks to my colleagues, really, I appreciate the effort they make, and thanks for listening. And maybe we can have a quick discussion. Do you allow us? Thank you.
I have two questions. The first one is more related to what you showed earlier. These spontaneous transitions between frequencies, this is the resonance results. Are you already at steady state when you simulate them on?
Well, yes. Yes.
Okay, so how long are you running the simulations for?
So
because if you're, are these frequencies periodic? Like, I'm not sure I understand this very well, to be honest.
yeah. Let's go there. So let's go even there. So all the nodes start at 40 hertz. All the nodes have 40 hertz. And then I set them to interact via their connectivities. You get that? Yeah?
Yeah.
Right. So if I let them do that, so we run it for a simulation of between 30 and 50 minutes. The simulation time, I mean, simulation time is days, but then we discard the first few minutes to allow it to get to its steady state.
Yeah, but I mean, if you're talking about spontaneous transitions, like I see the simulations that you're showing later. But for me, this is chaotic. This is not really like there's some sort of chaotic dynamics associated with that too. There is no regularity in the pattern. So how do you get this? And why would not be shown like this sort of chaotic dynamics be present within the spectrum?
Why do you say it's chaotic? There's nothing chaotic in the model.
So, for example, if you look at the first decoding, switching from green, like some frequency, to another which you did not label, to red, to green, to red, nothing, to green.
Yeah.
So this is not a regular pattern.
It's not, I mean, I'm showing you here a few seconds, but it repeats. It's exactly the same as you would have it in EEG. In EEG, we and others have shown that if you use something like a hidden Markov model, you will get a trajectory that repeats, a transition matrix you can estimate.
And you can take time chunks and do an FFT in a time chunk, and that's the spectrum.
It will look like that. But if you look over a longer period, these patterns repeat with a transition probability that you can estimate quite reliably. So a lot of the work was done by Mark Woodridge in Oxford and his team. We've published this in the same way to kind of estimate those states and their transitions and their trajectories. And you find that in the pattern of the trajectories, you can get information about what the person will be doing next with their performance in a task, in resting.
But this system is deterministic, though, right?
Yeah.
Yeah, but I mean, the EEG data there's noise associated with that, and that could shift you from one attractor to another, and that's why you get all of these, a whole spectrum of frequencies.
Correct.
But here, this is a deterministic model, and you're still seeing this transition.
Why in the EEG you still get a banded spectrum? In EEG you still get highly bounded spectrum. You don't get a continuous spectrum. If your argument is valid, you get a continuous spectrum.
Could push you based on, I mean, the noise can transition you between these different
This shows that you don't actually need the noise.
Okay, maybe I'm missing some details. We can talk later. I don't want to take
somebody hasn't spoken, so give somebody.
I had a question. What is the most important thing, the structural connectivity matrix or the time delays for the peak frequencies?
Beautiful question. Beautiful question. You can't separate the structural connectivity matrix from the delay, because the delay is a feature of the structure. The delay is the length of the fiber. So whether you say the structure in terms of the connectivity independent of the length, the topography, the graph property of the connectivity, whether that's more important or the delays, both have a role. If you change one, you'll get either of them will give you an effect. You can play the delay will give you the position of the frequencies. So because as we shown that the delay has a relationship with the dimensionality of the network, so the delay will shift the position of longer delay will shift these frequencies to be slower if you have a longer delay.
And I had a second question, is from the matrix you can infer some communities or subnetworks.
Yes.
Do you find the same local subnetworks as with here the E figure? The small networks on the local part? I mean, from the matrix you can infer subnetworks. Do you find the same subnetworks as in your simulation?
Yes, because you can see this in this is shown here. So here, this node, because in the graph, it is not extensively connected, it does not participate in global networks. It only participates in local networks.
Okay, fine.
Whereas the highly connected nodes have the chance to do local networks and global networks.
Thanks,
It's-
Wael.
it's-
Okay.
it's
And
not
we have
allowing you anymore.
yeah, well, and maybe you can discuss in the
Yeah.
break.
I'm here.
And now we have a 20 minutes break until 11:20.